So actually, the next talk is about quality management, QA, QC, that we need to do part of our research for the consortium. So Sean, you'll introduce the rest, I hope.
Yes, sir.
Okay. All right. So Sean joined the McGowan Institute in 2001 as the supervisor for the experimental surgery in the Center for Preclinical Studies. And then he assumed the leading responsibility for the development and implementation of the quality management systems program supporting the McGowan Institute. So the Center for Preclinical Studies and other departments within the University of Pittsburgh. So as the director of the quality assurance for the McGowan Institute, Sean oversees, implements, and provide quality assurance, monitoring, and regulatory guidance to investigator teams and industrial partners for exploratory and later stage research. So please welcome Sean and the team with him, who he will introduce.
The team. Yeah. Hey everybody, a little rougher QA, QMS talk right after lunch, so I hope you can hang in there with us. We're glad to see we have as many people as we do. We saw the bags stacked up around the room. We thought that by the time we got to the QA talk, things would empty out. So the first slide I want to open up with is the mission statement, an excerpt from the mission statement from your consortium, and an innovative toolkit access to exclusive mentorship and expansive community of translational resources. We're offering some resources here today from a quality management systems. Now, for about maybe 5 years, the three of us have been working together in developing quality management systems that fit in academic settings, which is really not that simple. But we've taken and leveraged other resources to find what works, how it works, and what we can make work. Oh, thanks. And what we can make work and what we can do to help our investigators and our science develop their technologies along the complete research continuum, from basic research all the way through to the submission to regulatory. One problem I have here is I had my notes. Can I see my note sections on this? No? Okay, so great. They improvise. So I'd like to introduce our first speaker from our group today, who I made her write her bio. Melissa Eitzen is from the University of Texas Medical Branch, where she works at the Galveston National Lab, where it's actually a hot lab. It's a biosafety level 3 facility. She works with the FDA, biosafety level 4 facility, where she works with the FDA in developing research and quality systems that works within that type of arena. Now, one thing I want to stress here, we all come from very different backgrounds, but we've developed processes that we would like to kind of offer and show you today that you can actually use as a resource to support your own science. So, Melissa?
I have 2 buttons and I'm going to get them confused, I'm sure. So thank you to the organizers for inviting me. Thanks, Sean, for including me. Thank you for staying. Full disclosure, my son is a 3rd-year student in dental school. And so I told him I was here. And so thanks for allowing me to be the cool mom again for about 24 hours, I'm sure. He was very interested in what you were talking about today. So what I want to do is I want to tell you about how we stood up a quality management program at UTMB based off of the fact that we're approving or we're helping do the research to approve medical countermeasures. So it's something completely different, but the concepts are the same. And we thought it might be interesting for you to take a step out of what you normally do and look at it through a different lens. So we had to use the GLP regulations as the reference for our quality system. You'll see why in a minute. But when I go into basic researchers, this is sort of the response that I get. Usually with a few colorful metaphors as they're saying it at the same time. So they say 21 CFR Part 58 GLP, this is nothing but a burden. This is inhibiting my research. Why do I care? Why do I have to do that? And really, if we take a look at the scope, which also tells us when these regulations apply, please understand that the intent of these regulations is to assure the quality and the integrity of your data. So it's not about the science. You're the ones that bring the science. This is the framework, the quality framework that the FDA uses as a lens to judge the quality and integrity of your data. And the reason is because, there we go. I'm sure you're familiar with this. Is overly simplified, the model that we use to get something translated from bench to bedside. But there's a couple of really big regulatory decisions here. One is when we first want to put a product, whether it's a medical device or a drug or a biologic medical countermeasure, into humans, and the other one is to get our permission to sell and to market this product. But prior to that GLP, which really applies to the safety toxicology studies, prior to that, there's no regulatory infrastructure, nothing standardized for us to really guide our research. So it ends up being voluntary, it ends up being patchwork, it ends up being haphazard, and unless, as we've heard today, you're employing somebody that has some experience with this, you can get lost in it very quickly. So why do we care about GLP down at UTMB? Well, first of all, it's a quality system, and there's a new GLP regulation that's proposed, a modernization of it, and the FDA even refers to GLP as an overall quality system to assure the reliability of your data. There's something called an animal rule. Does anybody here, have you ever heard of the animal rule pathway to licensure from FDA? Great, that's what I thought. So I can say anything and you'll believe it, right? So in 2002, FDA put in a regulation. They said there are conditions, there are circumstances when we can't ethically or feasibly do a phase 3 human clinical trial. If you think about the Ebola outbreak in West Africa, this is the best example I can give you, right? Over 28,000, 25,000 or so infected, over 11,000 fatalities. You couldn't predict that outbreak. You couldn't feasibly set up those phase 3 clinical trials in advance. And there was an ethical question in terms of how we would run those from a placebo-based trial. So when they're ethically and feasibly impossible to do prospective to market approval, then they allow us to do this in an animal model. And we have a large population of infectious disease researchers down at UTMB, and we also have one of only 2 national laboratories in the US, the Galveston National Lab, which allows us to do this work at BSL-4. And now we have a regulatory team as well. Perfect storm. So what they told us was these studies have to be done under GLP. And we said, but they're not safety, they're efficacy studies, and they're BSL-4. Oh, it's not a rat model that's a healthy rat model that we're doing toxicology. This is a disease model. We have to expose these animals to a challenge agent in order to test the efficacy. So this is a square peg round hole as far as we were concerned. So they matured a little bit in their thought process, and we helped with that, which was kind of nice because they engaged the community to do this. And they said, all right, but we recommend the GLP regulations to the extent that's practicable in order to assure, again, the quality and reliability of our data. And that really means that when they come to inspect our data, they're going to use the lens of GLP. So we have to know GLP, and we have to be able to explain GLP whether we like it or not, because that's the system they know, that's the next closest thing, and that's the measure they're going to use. So we have to be able to say, yes, we met GLP, or no, we didn't, and here's why, and here's how we mitigated that risk. But I would argue that it's not just required in regulatory. It's a really beautiful document out by the WHO called Quality Practices in Basic Biomedical Research. And they say that quality practices are really intended to increase the likelihood that, this is where you come in, provided the research has a scientific basis and the hypothesis is testable, research activities will generate reliable data, suitable for publication, and perhaps for further research aimed at detecting, preventing, or treating disease. And their philosophy really is that sound scientific principles in and of themselves aren't enough. You really need to partner that with good quality practices, and those together lead to the credibility and the reliability and the reproducibility of your results. If you look at this simplistically, it means you prospectively plan what your scientific experiments are going to be. Then you do them according to the plan. You capture the information, you report it in its entirety completely. You don't cherry pick the data. We also learn from our failures, by the way. And you store and archive these results because you never know when you may need to go back to them, and then you publish. And all of this is documented and monitored and or verified. It's the same for GLP. It's the same process. GLP is prospectively planned studies that are performed according to the plans. They have some independent monitoring, usually about 3 times. When you finally get to the final GLP studies, it means they're audited internally by the sponsor and then eventually by FDA. They're recorded and they're reported and they're archived. So they're the same approach. And what I like about this document is it breaks it down into what we call prescriptive and descriptive documents. So prescriptive are those plans that we're going to follow. It's our study protocol, it's our study plan, and our standardized procedures. Then we're going to do the study. We're going to have the study results that are going to be a bit nebulous depending on what the study design is. But the way we analyze and report that data should also be consistent. From their perspective, if you control this, I think it's a business perspective, quite honestly. If you control these pieces, then if you have 3 different studies that you get 3 different publications out of, you could effectively get a 4th publication because you could compare those 3. Because you've standardized the processes for how you do these in your lab. And if you think about the fact that you want to translate from bench to bedside, the sooner that you start being able to communicate this, the better it is. From a GLP perspective, that data is the deliverable. I would argue that that's what you're looking at too. You're analyzing that data and you're communicating it via publication or hopefully to translate it. But what really shores that up are all these pieces that are underneath that pyramid, starting with people. And you have to have the right people, you have to train them, you have to provide them with standardized procedures, you have to provide them with equipment that's well calibrated, otherwise you can't compare your data over time. You need to maintain integrity with your specimens, and you need to record all of your results and be accurate and complete in those results. I heard somebody earlier talk about using students. You know, there's a couple of things you can depend on in research laboratories. The fact that people are transient, right? And so that's one of them. And so you really need to think, well, how am I going to control for some of this and how am I going to become more efficient? Because that's the second thing we know is funding is becoming less and less. We have to become more efficient with less. So this is a nice diagram from the Research Qualities Association in the United Kingdom. And I really like it because it's those same things that you saw in the pyramid. You see if you've got people and training and equipment, monitoring processes, documents, and the facilities, where those standardize and overlap, that's really where you're focusing on your quality practices. Because those are the things you're going to use all the time, no matter what experiment you put in and out of that system. Those are the things that are within your control. And this is my last slide before I turn it over to Sean. And I wanted to say that at UTMB we used GLP because it made sense. We knew that our products that we were working on, we knew that they were going to go through that pathway, that we were going to be scrutinized by GLP. Might I also add that our researchers who weren't quite ready for GLP also got their data looked at, sometimes without them thinking about it, because there might have been an epidemic or a pandemic and their data was it. And NIAID funded them. There's a flu epidemic. We know Dr. So-and-so has got an animal model for that. What data do you have? FDA wants to see it. And we never know when that's going to be. So we're good stewards to be able to prospectively think that what we're doing, even in research, might make a difference at any given time. And so this is the environment within which I work, and this is why our management supports us. But it doesn't have to be GLP. If you think about what it is that you're trying to translate, it could be the ISO 9000s, it could be 15189 if you're looking at clinical things, it could be WHO if you're looking at some global things. But when you do that, you really do define the gaps in your program and you can manage those risks prospectively. And that's the whole point. As scientists, we like to be in control. I'm telling you, this is another method where you can be in control. And I guarantee you, we see it with the NIH and their rigor and reproducibility initiatives, and you see it in the decrease in your % funding. Because the money is getting less and less and less, then those expectations for some kind of assurance on the quality are going to be increasing, and they're going to look at what's already out there. So think about that. Leverage your QA professionals. We're sometimes fun. We're not all bad. We're not always a wet blanket, and we're always here to help. So I'll turn it over to Sean with that. So thank you for your attention, and we'll take questions if you will.
So Melissa gave me a good segue there into rigor and reproducibility expectations. The increasing expectations by NIH and other funding agencies that we have to keep in mind. But what does that really mean, rigor and reproducibility? Well, we have to really look at it from the sense that, quality assurance does come in here. A quality management system starts way back in the earlier stages of your science. They really, they do go into rigor and reproducibility, what the expectations are, but they really don't tell you how to get there. There's really no map. And that's where we argue the quality management systems play an important role at this stage. So there are two specific areas commonly referenced. We're seeing coming back from regulatory feedback from the FDA. This is the post 60-day time point in which the FDA has responded to a pre-sub meeting. And one of the things we're seeing, and pardon the redactions, I especially like the one that says "and" right there in the middle. I can show you that. But this excerpt that I've taken from this is they're highlighting two areas. One is 21 CFR 58120 and 21 CFR 58185. That is 58120 is your protocol and how you conduct your study, and 58185 is your report you provide to the FDA as a result of your study. And so these are those two sections, subsections within the GLPs. One thing I'd like to point out, in fact, what we've done is we boiled this down a little bit to make it a little more friendly so you can take a look at it. They basically say the same thing. The protocol tells you, the protocol pretty much tells what you're going to do, what expected you to do when you're performing your science, your study. The report is what you've done and how you've conducted it. And there's a lot that goes into that, and it's something you really need to pay attention to. And the other point I'm going to make about these things we're seeing from the FDA is that each one of these, 21 CFR 58120 and 185, both imply the rest of the GLPs. In other words, you have to really follow the other parts of the GLP regulations to meet these two areas. So the expectations from the FDA is pretty much GLP. That's FDAEs. That's the language they speak. This is how the FDA wants you to communicate your science to them so they can make a decision. So just going to kind of go over quickly of the two areas, the experimental design part and the protocol key elements, experimental design. These are the primary area where they're looking at records, the records they expect you to maintain, type and frequency, test analysis, measurements, statistical methods used, so on and so forth. And then, of course, we move over a really important part is your animal models, how many you used. Believe it or not, how you identify your animals throughout the study is a key element. They really look very closely at that. Dietary information, is there anything that's influenced, especially some of the things that you guys are testing, or is there anything that specifically that may influence the outcome of the study? You got to keep that in mind. And then, of course, the characterization of your test article. That is huge. That is something you have to be well defined, and a lot of things get kicked back because of poor characterization. Administration, application, implementation, description, how you went about your study. It's well described in your protocol. And then we move on over to your final report. And there are two key areas here that I think that I need to point out as far as your final report, which are here, a description of all circumstances that may have affected the quality and the integrity of the data. And then, of course, over here, quality assurance statement providing dates of inspection and date of findings reported to your management. So the FDA says, yes, please follow 58120 and 58185. What they're essentially saying is follow the GLPs, and you have to really have these elements in place. Again, the final report, stability of it has to describe the stability of your test article under the variety of conditions, route of administration, objectives and procedures as stated in the protocol. You have to make sure that you clearly define that. Again, more about your test system or your animal model. That's a really key part of all this. You really have to make sure that you provide all the information as required by the GLPs. And of course, statistical methods used for analyzing your data. How did you analyze it? What did you use to analyze it? And so this is just a graphic we put together that I wanted to try to demonstrate that how quality management really is actually in play already and how it supports throughout the entire research continuum here. So you have your exploratory, which is generally, we're all in academia, we have an IACUC protocol that's approved. That's essentially something that does deploy some quality management systems, you're reviewed, you have to, any changes to that, there's change control, you have to get modifications approved and so on and so forth. You have to document what you do with your research animals. And then you move on, and we're going to move on to this area right here first, and that's the non-clinical study protocol, which is the 58-120. This is compulsory, required by the FDA. You have to make sure you have this in place and all the elements as we previously discussed. But then in between that, there's an area right here that's study requirements document. Now that's something we've adopted in our quality management system at the University of Pittsburgh's McGowan Institute, our Center for Preclinical Studies. And what that is kind of a study protocol, but it's information taken from your ICUC protocol, but you've also added, a variety of elements that may not be in your IACUC protocol, because a lot of people tend to use their IACUC protocols as their guide to how they're going to conduct their studies. And I know a lot of personnel do within labs. They'll go back and check the IACUC protocol. But an SRD or an ERD, a study requirements document, can provide that gap that really helps you carefully plan your study, that helps you identify critical processes that may need to have additional SOPs. It will help communicate what you want to do with your staff, whether you have 1 or 2 people in your lab, whether you have 10, 15 people in your lab. This is an opportunity to really help translate what you want to do to your personnel, but it also helps you think critically about how you're going to conduct your study. This helps reduce the potential for redo. Redo will kill you. If you have to go back and you have to redo something because you didn't plan it carefully, or you forgot something, or you didn't have the right procedures in place, or the personnel weren't appropriately trained, this type of a document will really help you out. And it's helped out many of our investigators. A lot of them take it, kicking and screaming, and other paperwork and all this other stuff. But I'll tell you, towards the end, they're really happy they have that. And as a side note, anybody that might be interested, as a resource for this resource center, I'm happy to provide you a template for it. It really helps make a difference early stages of your research. So these are the other type of things that we put in there. And it can, one thing I want to point out to you, not that you're going to be overwhelmed by documentation, it's 15 pages long, big deal. And that includes, personnel, roles and responsibilities, who's going to do what. A lot of it's taken from a GLP type protocol in 58-120, but the majority of it's kind of light. It's kind of helping you define and work through your study or your studies. So, and there are other resources to help you build your internal quality management systems. The SRD is one. Developing of standard operating procedures for your critical processes is another. There's a lot of information out there. One thing about the GLPs that you have to keep in mind, they assume that you have the basic foundations for a quality management system in place already. It's kind of a QMS in flight. The other options that we have here, as Melissa brought up, was the quality practices, and basic research that WHO put out. Another good resource, though they're kind of expensive, are the ISOs, ISO 9000 and ISO 9001, where they actually provide you the terminology and somewhat of a framework that you can take and adapt to your system that you may want to develop in your lab. And of course, other documents that are out there. So if there's anybody that wants to reach out to us, and if they have any questions about developing an internal QMS, I would strongly recommend you do so, because it's one good way to really protect your science early in the stages of your research. And with that, I'd like to bring on, oh boy, here we go. I don't have my notes up here, so now I have to give him a... This guy is Mike. So Mike is the Associate Director for the Center of International...
Close enough.
Okay.
International Regulatory Sciences.
International Regulatory Sciences at the University of Southern California. Mike?
And the best part is Sean made us all write bios so we could read them, and he didn't actually have them today, so that's a bad sign. Just quickly, I was in industry for 30 years before I came to academia, so at lunchtime Sam talked about going the other way and all the flack he took. I think I probably took more flack going the other direction, especially from my wife, who, as she said, I cut my salary in half and doubled the amount of time I worked, so she's still very excited about this transit. But I came to academia to be able to do things like this. So when Jeff Lotes approached me about being involved in C-Doctor, it was probably the most exciting things that's happened to me as part of my time in academia, because I came to academia because I believed, or maybe naively believed, that we could change the way the system works, whether that be tech transfer, whether that be quality of data, reproducibility of data. But when I was in industry, one of my responsibilities was to work with academic researchers on projects that we were looking at funding. And it could be one of the most frustrating things to deal with, because I would go into the lab and I would look at their data, and the first thing I would do, because I was trained in more of an FDA way, is I'd say, well, when was the last time you calibrated your HPLC that you used for your PK/PD data? And they'd all look at each other and someone would go, do you know? Do you know? And it might be 8 years ago. And that doesn't mean that the data was bad, but my sense or my comfort zone with that data all of a sudden changes. I'm a little bit nervous about that. So again, I came to academia because I got to know a lot of researchers who were also frustrated with the system, and I think there's a way to improve this whole translational process. And part of that, or where it starts, I loved what Jeannie said about this. It's never too early to start. You can't just do it step by step. You have to know that almost every project, I'm the resource director in the California Sea Doctor, and it's one of those cases where every one of our projects will go through GLP at some point in time. So you know you're going to get there. That doesn't mean you have to do it now, but you know you're going to get there. So when we talk about this idea, and this is again right from the webpage, from the Sea Doctor webpage, the idea of accelerating technology translation. Whenever we put the word accelerate there, that always gets people a little nervous, and they all want us to start talking about what we mean by accelerate. Sean touched on it, and what I see as accelerate is that not having to do the same thing twice, not doing the proverbial 3 steps forward, 2 steps back. So if you do it right the first time, you don't lose that time. So if we're going to accelerate that time from bench to bedside, it's very important that we don't have to keep redoing things over. And more importantly, our potential corporate partner doesn't have to redo things over again. It adds value to our technology if we can actually be seen and giving them the confidence that they don't have to redo all our results all over again. And what we talk about is, and this is sort of the statement that I think is probably most important, I think is really to the crux of what we're trying to do in Sea Doctor, is if you cannot reproduce your research results, you will not be able to translate that technology. You will have a very, very difficult time finding a commercial partner. So not only is it from a good science standpoint, but it's also going to increase the chances of you commercializing your product, but also the speed in which it's going to be translated. This is, again, I spend a large part of my time talking to venture capitalist companies. I'm on my 5th startup now. And the reality is, for years, even before all the issues with reproducibility came out and NIH started to address it, there's always been that concern about the reproducibility of academic data. And so a number of publications have talked about this. It says at least 50% of VCs ask for some type of external validation of the data as a milestone before you get all of the funding that you're going to ask for. So reality is there's always been this concern. But some schools, some researchers are better at than others are. So a lot of the successful researchers, industry trusts their data. We've all talked about this. Again, it's been in industry. I always started in industry in 1984. We've always had this concern, but it's really come to the forefront in the last 5 to 7 years. And the biggest one probably is the one you've all seen, the one that was done by Amgen, and it's this study right here, where they talked about only 6 of 53 papers could they reproduce. Now, we can sit here and argue for the next 2 hours about the pitfalls in another entity trying to reproduce research results. I actually talked to Begley and said, well, did you actually go back to the researchers? He said, yes. We went back to them and said, can you help us figure out why it's not working? So again, whether this is true or not, perception is reality. And the reality is 11% was reproducible. So I would say now that this is one of the most well-known papers from investors, from angel investors, from venture capitals, from industry, is the concern about this reproducibility. So it's become more of an issue. And I would guess that this is part of what forced NIH to look at reproducibility and rigor, was this became more in the public domain. So again, we look at this, NIH over the years has talked about this, and they themselves have addressed the issue that the system for ensuring reproducibility has some issues with it. So again, it's been identified by the funding bodies. Also talked about what stage it was most applicable to, preclinical research, which is what we're talking about today. That's the area where a majority of these issues are occurring. And last but not least is this idea of training. So this is, again, where you've heard about it from C-Doctor here in the East, and what we're doing in the West is part of C-Doctor is to train people so they understand how this works, what the process is, what the pitfalls are. So we're trying to address those as part of the C-Doctor resource centers, is to try and address these issues of training and understanding. Jeannie talked about this, when you look from an industry standpoint and an academic research, one thing that industry sees that academia doesn't understand very well is the time for things to happen. We did a survey of all the major drug, device, and biologic companies in the United States and asked them what they found were the goods and bads about working with academia. The number one was the bureaucracy of the system, but the number two one was the lack of understanding of the overall development process, the cost and the time, and where your technology was in the development cycle at the time you were trying to convince a partner to invest in it. So again, understanding this cycle is really, really important. So last couple slides here. This is an article that was written, and this is the discussion that's going on right now, is who's responsible for doing this? Should it be the funding agencies? Should it be NIH? Well, NIH has started to address this in their grants. You all know that in the applications, this idea of rigor and reproducibility. Should it be the journals? Well, we know journals like Nature now, in some cases, are asking for raw data so they can crunch that raw data to ensure that that data is of a level it should be. Then we talk about the institutions and the researchers. Should the institutions have a responsibility to help with this? That's what I'm trying to do at USC. I'm trying to get us to implement a system. Simple things like a school-wide calibration system internally, so that researchers have the access to calibration of their equipment, not something you have to put in their grants. There's obviously a cost benefit by having multiple researchers all using the same system, but is it an institutional responsibility? And when we put this together, that's why Sean and Melissa, I did it this way, because I believe that UTMB's system and the system here at McGowan are probably the two best systems at US universities right now. But they're not institutional wide. These are small entities that are very successful in what they're doing. And I think if I talk to somebody like Patrick, who's sort of involved on the attraction of commercial partners, he would say there's been a benefit from having a system in place and giving that assurance to the industry partners. So again, there's definitely a benefit here. So the last slide, and again, what C-Doctor is trying to address is the fact that one of the best ways to address the issue of reproducibility and quality of data is some form of quality system. But as Melissa said, when we walk into a lab and we say the word, anything that starts with the word good, PIs want to run the other direction. There's no doubt about that. So that's what we're trying to do, is develop something that's not a regulated quality system. Basic stuff, things like SOPs, good laboratory note-back practices. We're not saying validate your whole methodology, but the critical parameters. At lunchtime, Sam talked about CMC, the whole idea of chemistry manufacturing. If you have good control of that, when you pass it over to that contract manufacturing organization that's going to do it for you, that's probably going to go faster and probably smoother and probably less expensive if you have done a good job of documenting your critical steps. So there's a benefit to that. But in the end, what we're talking about is the fact that in the current status of the system, PIs in most cases are on their own. Individual PIs are seeing the importance of doing this. I have a number of labs at USC that are doing it. Charles is a good example, who's trying to do it in his own lab here at Pitt. But there's really nothing that's been formalized or standardized. And I think that's one of the opportunities that we have in C-Doctor, is to try and develop a system that is very flexible, not overly complicated, not going to affect the science, but allows as a group that when we do try to attract commercial partners, that they have a level of confidence in the data coming out of the C-Doctor grants, that they feel that they can trust the data coming out. And I think moving forward into phase 3 of the project, that will hold us in great stead when we reach the commercialization stage. That's it. Any questions? It's always nice being the last speaker on the last day, and they have to get on a bus at 3. So that may have been the fastest I've ever spoken in my life.
Hi, could you discuss GLP Light, the FDA's stance on that, and then also what level of GLP can the course support, full GLP or something less than that?
Okay, so I can answer that. The FDA will say it's either GLP or it's not. And I can tell you from our perspective, it's better to understand the GLPs and then self-disclose where you're not GLP. They consider that a risk because that's the minimum standard. So you're not meeting the minimum standard. So then you have the ownership of saying, well, if I didn't meet that, why? And how did I mitigate that risk? So for us in the BSL-4 environment, it's more easy for them to understand and appreciate that there are some risks. The space, even though we have 12,000 plus square feet of BSL-4 space in our Galveston National Lab, that space is of a premium, and we have basic research right next to what we would consider regulatory research. There's a risk there. So we just disclose that and mitigate it. But and even if we don't do it GLP, these animal rule studies, and we say we're not doing it GLP, but here's where it is, the inspectors whom I know and are part of faculty in a course we run will tell you they inspect a GLP and they write 483s, whether you say it's a GLP or not. That's their standard.
I want to add to that too. A lot of what we're seeing coming back from the FDA, I've seen it 3 or 4 times over the past years. They reference, especially for devices, 812.27 and 814.20, I believe it is, where you discuss whether or not you've conducted your studies under GLP. If not, provide a brief explanation as to why. Now that a lot of people are making the mistake thinking, okay, this is my get out of GLP card. It's not. You just have to be able to communicate to the FDA why you didn't make certain parts of, comply to certain parts of the GLPs. So be very careful with that. Exactly. You have to disclose that. That's the last line, and that's it. Yes, you're absolutely right. What's the impact?
This is a little bit out of the main focus here, but in terms of relevance to the startup companies. So we're hiring a CRO to do biocompatibility testing, 10993, and they go, here's your quote. Oh, we don't have to do this GLP because CFR says we don't have to, blah, blah, blah. Oh, and if you want to do GLP, it's going to take longer and cost you more money. So we really want you to take this quotation. And so who do you believe? The CRO multi-million-dollar worldwide testing lab? Or, I mean, where do you go with that?
If it's going to be submitted to the FDA, it does have to be done under GLP. And they're telling you 10993s, and there's guidance to 10993s, but that's been kind of wishy-washy as well, so you have to be careful with that. I would be very careful. I would assume that, as Melissa pointed out, understand and work toward the GLPs. And if there's something, especially when it comes to medical devices, if there's something you cannot meet those GLP regulations, if you can articulate that up front, then you might be okay. But if somebody's telling you, well, you don't have to, as long as you follow 10993, you're okay. I wouldn't buy into that. I'd be very careful with that.
All I'm just going to add quickly to that one is we had this discussion. Part of my responsibility in the West Coast Sea Doctor is to vet those for you. So because that's, as any of our ITP teams, I will actually go and visit and have the discussions with any of the CROs or the manufacturing facilities. So I'm guessing there's somebody here in the East that could help you with that, because just trusting them 100% is not the right thing to do. Yeah.
Amen.
Well, thank you. Well, thank you all. I know everybody has to catch a shuttle. I'm just going to leave the closing remarks to Dr. Cohn, and have safe travels.